hope to cover today, maybe give you a bit of perspective about myself, and I won't get too much into it. Talk a bit about the value of a chemical engineering education, some of the careers that you will have in chemical engineering. And since I am I work at Pfizer, I will provide you with some perspective from biopharma and what I do as a chemical engineer at Pfizer and what others do as a chemical engineer at Pfizer. And then maybe conclude with my thoughts on the chemical engineering department at UVA, and then we'll have Q&A after that. I will skip this one because we don't have too much time. So just so my affiliations, can you guys see at least what I'm showing as we're going along? Okay. In terms of my affiliations with chemical engineering department, just to be clear, I am a graduate of the department. I got my PhD there in 1999. But I've also been on the Chemical Engineering Department Advisory Board since 2003. So except for the last 2 years, because of COVID, I have had the fortune of visiting the campus every year, which has been something that I always look forward to each year. So let me put this a little bit. One of the key points that I want to make is that chemical engineering education really provides a strong foundation for advanced degrees in a diversity of fields, and that is really important. I think it's to me it's somewhat unique in chemical engineering that with that foundational background, there's so many doors that are open up to you. First, after you get your BS in chemical engineering, you may choose to go into industry, and a number of people do that. You may also choose to go into graduate school, which is what I did, and a number of folks do that. But with chemical engineering education, a number of people also choose to go into medical school. In fact, a friend of mine from undergrad is a nephrologist and went straight to medical school after undergraduate. Others also choose to go to law school, and you can either do patent law or any kind of law. And others, at times, after working for a year or two, usually choose to go on to business school. And so I think the key point that I'm trying to make here, that the teaching you get in chemical engineering, the foundation that you get, it doesn't close the door to you to just the chemical engineering field. In fact, it opens the door to you in multiple fields. And that to me is very unique in chemical engineering. And that's why I like chemical engineering very much. You guys can see it okay, or do I need to move it a little bit? Let me see if it's presentation mode. Okay, I put in the presentation mode.
Mine is here actually, if you would like.
Are you?
Yeah, I can share my screen.
Okay, all right, and then you can go on slide 7. Mm-hmm. This is not going to be the best recorded one because there's so much interruption, so I apologize for it. All right, you can just jump to slide 7. That would be good.
Mm-hmm. Wait 1 second. This one, right?
Yep. Okay, let's see. So with the chemical engineering degree, there's also a broad and diverse industry that you can participate in. And I think that's also very important. Obviously, the word chemical, so you can always get into the chemical industry. There's also energy, and it's a huge area of work, as many of you will know. Clean energy, oil and petroleum energy, and nuclear field. You will always see chemical engineers in any of those fields. Chemical engineers are in the semiconductor field as well, in the food industry, materials, mining, environmental, biological, biotechnology, and biopharma. Now, you can, for example, have someone who's a nuclear engineer, but you will never see in a chemical engineering field. But you can have a chemical engineer be in any of these fields. Again, it speaks to the really the diversity and the broadness of the education that you have in chemical engineering. So again, the point here is that chemical engineering opens significant doors for you. I'm going to take the next few slides and really focus on the biopharma industry, because that's what I'm most familiar with, and maybe give you a flavor of the company I work for, Pfizer, and some of the things that we do within my company. Next slide, please. So in biopharma, there are really two main areas of research that we do in biopharma. One is what we call small molecules, which typically are chemistry-based drugs. They're really well-defined drug, right? So it's a small molecule thing. And within that area, there's also process development and manufacturing. And chemical engineering, in addition to the being a big part of the whole organization, chemical engineers, you can see a lot of chemical engineers in these areas. Okay, chemical engineers are involved in the development of the process and then in scaling it up to commercial scale, and also in manufacturing it when the drug is approved. Similarly, the other part of it is what we call the large molecule, biologics-based thing. Again, it's the same thing. You have process development and manufacturing in there, and you see chemical engineers as the big part of that. And so at UVA, you will get either bio focus or chemistry focus or many of the other focus, right? And so for pharma, for the biopharma industry, UVA prepares you really, really well for it. Next slide, please. Now, what is a biotherapeutics, right? I'm trying to kind of put it in perspective and give you a sense of why we call it large molecule. On the graph on the left, you have aspirin. That's a small molecule chemistry-based drug. It's typically very small, in this case about 180 Dalton, and it's really well defined, right? You know the actual structures of them, so you can basically define what structure that is. As you get into the biologics, from insulin to EPO to things like monoclonal antibodies, they get larger. And you can define the primary sequence of these molecules, but there are lots of post-translational modifications that are made on top of them that make them very unique, right? So there are sugars on top that really could impact the activity of that protein. And I have on the right some pictures that shows, schematic of various biotherapeutics. You can have antibodies, and many of you may have seen commercials for like Keytruda or Humira on TV. Those are antibodies for cancer. Some of them are conjugated with a drug to make them even more effective. There are various versions of that in bispecific fusion proteins, vaccines, nanoparticles with proteins inside of them, and gene therapy. So the area of biotherapeutics is wide and expanded, but is in essence anything that's made with biology or cells are considered biotherapeutics. Next slide, please. So a bit about Pfizer, and this is a bit outdated. It's from 2019. At Pfizer, really, our purpose is really to develop breakthroughs, medicines that really change patients' lives. It's a global company with about 88,000 people. We are in over 125 countries. We manufacture in about 49 different sites. Now, we measure our impact on society by the patients that our drugs impact. So in 2019, for example, we vaccinated over 69 million people. This is pre-COVID. I think if you take 2021 into account, our impact will probably be billions just on the COVID vaccine. Okay. But we have research in multiple areas. But I think we measure what we bring to society by how many patients really benefit by taking our medicines. Next slide, please. We have research in multiple areas. The key ones are in oncology, internal medicine for things like cardiovascular disease. We are big in vaccines, as you know, inflammation and immunology. You may have seen our commercial for Xeljanz, which is a small molecule inflammation drug that we make. And we also do research in rare disease. Now, as a chemical engineer, these things are really, they don't matter, right? You work on drugs, you develop drugs and proteins that have the potential to impact all of these areas. Some of the biology will be focused on oncology, internal medicine, vaccines, but as a chemical engineer, you work across the board, and what you do impacts patients across all of these research areas. Next slide, please. Now, my group is what we call a biotherapeutics pharmaceutical sciences. It's a global group with about 1,200 people. We have sites in Andover, Massachusetts, Morrisville, North Carolina, Chapel Hill, North Carolina, Chesterfield, Missouri, which is where I'm based, and even Cambridge, UK, where we do our devices work. Again, our focus is on large molecules, and we really develop the processes across the board on any biological vaccine product that Pfizer makes. Next slide. So enough about Pfizer. Now I'm going to tell you some of the things that we are doing that are so exciting that I think chemical engineering education are really critical for them, right? And that's the evolution of biotherapeutics and vaccines. And really, it's about from protein to DNA and RNA, right? So, I'm sure many of you have taken biology, but in essence, what you have for anything is you're going from a DNA to RNA to protein. With DNA, you transcribe it to RNA, and then you can translate from RNA to protein. Traditionally, our medicines have been mainly focused on protein, making proteins. But recently, with advancement in genetics, now we are making drugs that are either based on RNA, like we have for the COVID vaccine, and I'll touch base on that a little bit, or DNA, like we have for vaccine. Now, all of these things are essentially making the same thing. It's just a function of where you start and what delivery system you use to make it. Okay, and just to make things simple, I'm going to try to kind of explain what each one of these are and maybe use a few examples of what chemical engineers do in trying to make these. So next slide, please, Gino. So let's say you are making a product for a hemophilia patient, right? And if you know hemophilia patients are patients that typically are not able to make the anticoagulant proteins that prevents them from bleeding. So if they have a little cut, they could bleed a whole lot, right? And so in some of those cases, you have to give them therapeutic proteins. And currently, the main treatment is protein. And you give them, for simplicity, let's say factor 8 or factor 9 or factor 10, depending on which protein they are lacking. In fact, Pfizer makes one of them for factor 9, which is called refactor. Now, how you make that typically starts with the gene for the protein. So you have that gene, and then you insert it or transfect it into a cell. And for most mammalian projects, what we use is called CHO cells or Chinese hamster ovary cells. Then once you transfect this gene into a cell, you can expand it, right, into multiple cells, put them in small vials, right? And that really is called master cell bank. That becomes the starting material for whenever you want to make that protein. Right? And so when you're ready to make the protein, you take the cell, a vial of the cell, you expand it, you put it in a cell culture, a bioreactor. And there are significant chemical engineering effort here. So you have cells, you have nutrients, you have mixing, you have oxygen, and ensuring that the cells are in the right environment to be able to make the protein of interest with the highest yield involves significant chemical engineering principles, mass transfer, okay, a key part of that. Once you make the cells, once you grow the cells, and the cells will make proteins. Now, they don't just make the protein that you want, they make multiple other proteins that are typically secreted. In some cases, they are intracellular, in others, they are extracellular. In this case, let's assume it's an extracellular protein. So you make the protein, it is secreted. Now your goal is to isolate this factor 8 protein from this bunch of cell culture means that you need separation sciences. Okay, again, material balances, mass transference. So you have to go through a series of purification, either a chromatography, a filtration. All of those involve chemical engineering principles. Then once you purify the protein of interest, in some cases you may do some conjugation to it, in other cases not, but then you formulate it. And formulation into vials also involves some chemical engineering principles, because what you are trying to do is put the drug in a state where it's as stable as possible. So at the end, you have something called factor VIII, the protein that people with hemophilia don't have. And so you will give it to them. And so they will take it, sometimes weekly, sometimes multiple times in a week, to ensure that when they have a cut, they don't bleed to death. So that's been the traditional way of treating hemophilia patients. Next slide, please. But what if you can actually give them the gene for factor VIII, such that the patient can make factor VIII on their own, right? So instead of giving them the protein, give them the gene. And if you give them the gene and it's inserted into the nucleus of the cells that you want, the patient can then make their own factor VIII and would only require one dose of this gene given to them. This is the new area of work that we call gene therapy. And there are really two main areas. The first one, and is on the left side, is what we call ex vivo gene therapy. In which case you isolate cells from a patient, you insert the gene, and typically they use lentiviral with the gene of interest in there into the cells. You grow them in flask, just like you were growing proteins in the bioreactor. The genes get into these cells, and you give it back to the patient. This is called cell therapy. And it's a type of gene therapy called cell therapy, and it is used to treat lots of cancer patients. The part that I'm going to focus on the most, which is what Pfizer is involved in, is the one on the right side, which is in vivo gene therapy. In this case, you are making the gene and putting it in a virus, which we call adeno-associated virus. And you have this AAV with the gene of interest in there. You make that in the lab, just like you were making the protein, and you give it to the patient. Okay, that's in vivo gene therapy. The exciting thing about this thing is, again, it's a curative treatment. Yeah, you're not basically giving protein where they're taking it weekly. If it works, the patient will be able to make the protein on their own. Now, the science is exciting. How you make it is extremely challenging, and that's where chemical engineering principles come in. Next slide, please. So to make gene therapy, and this is just one form of making it, we call it a triple transfection thing. First, you need to make 3 plasmids. So you have to go and make 3 independent processes to make 3 plasmids. Now, I'll explain to you in a bit. Check the time. First is the plasmid for the transgene, and the transgene is the gene that you're really interested in making sure that you give it to the patient. And then, depending on the serotype of AAV, there's a rep and a captain. That helps you make the viral particle, right, in which the transgene is inserted. And then you have a helper plasmid, which basically helps to put the packaging of the AAV together. Now, what you have to do, the reason why we use something called transient transfection is because some of the genes in the helper plasmid, we call the E2, E4, are usually toxic to the cells. And in this case, we have 293 cells, human embryonic kidney cells. And so what you have to do is, first you have to mix all of these plasmids together, very time sensitive, and then you have to transfect it into the cell. And while the cell is making the particles, the cells are also dying because the genes on the helper plasmid are killing the cells. And then, within a short time, they have to make these particles. And if you're lucky, most of the particles will have the gene in there. But the reality is, a lot of the time, you perhaps have more particles without the gene. And a big part of your job, in addition to making sure that you can produce this particle, once you make it, is to purify it such that the product you have at the end is mostly the particle that's filled with the transgene you want. It's a complex process to make it, and chemical engineering principles are critical to making these things. Now, sometimes it's easy to make it at small scale in the lab, but to then scale it up and make enough for patients is hugely difficult. But with chemical engineering foundation, you can play a role in getting this therapy to patients. Next slide, please. So there are actually, this is a new area. There are actually 2 approved therapy that I want to tell you about. One is one for the eye. It's called Luxturna, and it's made by a company called Spark Therapeutics, which is now acquired by Roche. And in essence, what you do is they have this AAV particle with the right gene in there, and they inject it into your eye. Now, these are people who have this monogenic genetic defect that in essence will become blind. Okay, but with one shot of this gene therapy, they're able to make the protein that they need to be able to see again. Okay. The other one that perhaps you may have heard of is called Zolgensma. And that's a gene therapy for a disease that is called spinal muscular atrophy. And it's usually in kids. Typically, if you're born and you have that defect, you don't have that gene, or that gene is defective, you get into wheelchair, and within a very short year, if there is no treatment, there is a protein treatment that Biogen makes, but if there's no treatment, typically they end up dying at a young age. But with this drug, right, you can give it to a patient one shot and you overcome their spinal muscular atrophy issue. Now, this is a huge deal. This is the most expensive drug out there. It's $2.1 million per dose. Now, I don't want to get into the ethical part of it, but you can have treatment for your whole life, or you can get one shot and be cured for the whole thing. It brings some interesting ethical questions that I think it's going on around gene therapy. But the most important thing, really, as a chemical engineer, you are this is, I mean, I think the scale up of this is really the most critical part of the whole thing. And as a chemical engineer, you are able to play a role in such things to be able to develop these medicines that have the potential to cure people. This is a new area, really exciting area, and you can do that with chemical engineering. Next slide, please. How about the COVID vaccine? I think that's very much on people's mind. And again, at the bottom, I just want to go back to that, the dogma of DNA to RNA to protein. Now, many of you have heard that there are multiple vaccines that have obviously been approved. Emergency use authorization have been given to them. All of them are, in essence, targeting what we call the spike protein. And on the left side, you see a 3D model of the COVID-19 virus, right? The SARS-CoV-2 virus. Now, what these viruses have is they have what you call spike proteins on top of them. And these spike proteins are really the key to get into your cells. So when you get when you get in contact with the virus, it uses that spike protein, and there's a receptor called the ACE2 receptor that it then goes and basically opens it. And once it gets into your cells, This is an RNA vaccine virus. It lyses, releases its RNA gene into your body, and it starts replicating, and that's when you get COVID-19. So all of the vaccines in development are really basically being developed based on this spike protein. So when you get the vaccine, you are not getting the virus with its RNA. The goal is to get your body to make the spike protein, so then you can build immunity to it. Now, there are multiple ways of doing it. Again, it goes back to the DNA dogma. And J&J and AstraZeneca use what we call non-replicating adenovirus vector. Now, this is different from what I talked about in the gene therapy, where it's adeno-associated virus. This one is an adenovirus, and it's a DNA virus. It's basically what they do is they put the DNA of the spike protein into this adenovirus, and the adenovirus is really the carrier to the cells of interest. Now, I should point out that this is not permanent. It doesn't integrate. It's non-replicating, unlike the gene therapy part. But what the J&J and AstraZeneca vaccine is really doing is it gives you the DNA, and then your body will transcribe the DNA to RNA, and then it will then translate it in the cytoplasm into the spike protein. Okay. There's a company called Novavax, which is actually making the protein itself. That one has not been authorized yet, but I think it's pretty close. And basically, it goes back to what I was talking about. Their vaccine is basically making that spike protein, and they use insect cells for that. The Moderna and the Pfizer vaccine is the intermediate one, right? They use the RNA for the spike protein. So what we actually do is we make the DNA is a template. And then we use transcription, again, growing it in a reactor to make that RNA portion of it. And then because RNA is inherently unstable, we put it in what we call lipid nanoparticles. That protects the RNA. And that's what is given to a patient. So in that case, you are getting the RNA, it goes into the cytoplasm, and then it makes the protein. So all the vaccines are essentially the same thing. It's just the starting point, and the carrier that they use is what is different. Next slide, please. So how does the RNA vaccine work? And this is basically just a schematic of it. In essence, again, that green thing is the nanoparticles, and you have the RNA inside that nanoparticles, right? Once it gets into your cell, it releases this messenger RNA, and then it is translated to the spike protein. And that's when the immune system reacts to it. So this is a whole lot of immunology. I don't fully understand it, but the way it works is that the protein is then on an antigen-presenting cells. Your immune system respond to it through the T cell and the B cells, and the B cells ended up making the antibodies. Now, the B cell and T cell are really important because they basically what you call adaptive immunity. It helps your body remember how to make it, right? And so the antibodies are the neutralizing, will neutralize the virus if you get it, and the memory cells will help it keep it longer. And so in essence, the mRNA vaccine basically goes from mRNA to the translation, and then the mechanism thereafter is the same. The DNA vaccine, it's slightly different in that it starts a little bit upstream of that, and then it goes through the process. And the protein vaccine is, in essence, you make the protein and you move forward with that. So I hope all of these things, I think, well, let me show one more slide. Next, I think I have one more slide on this thing. But I hope what I've captured really gives you a flavor of what chemical engineers do in the biopharma industry. Because making that mRNA vaccine is very unique. Again, making it one thing and making sure that it's stable is another, and chemical engineering principles are applied throughout the board. So I think this is my last slide, and why UVA Chemical Engineering. First and foremost is that the faculty is excellent. You have faculty who really care, and the student to faculty ratio, it's very robust. I think it's one of the best in engineering schools. You will not feel lost in the Department of Chemical Engineering. So excellent faculty. Second, there is a strong focus on undergraduate teaching. I think that's been a stronghold of the department, and almost every year, someone from the department wins some type of a teaching award. And so that's really good for undergrad, especially when you're trying to really understand the fundamentals of engineering. There's also diverse faculty research in multiple areas. Again, chemical engineering is broad, and so there's that diversity exists in the department, and there are plenty of opportunities for undergraduate students to do research in any of these areas. And so if you are one, like I was, interested in the biomedical, even if you're interested in biomedical, I mean, there's biotechnology and biomolecular engineering is a big part of the chemical engineering department. So you need to go to biomedical, you can do it all in chemical engineering. But you can also do energy and the environment, and that's a huge growth area, as many of you know. There's materials and nanotechnology, and then simulation and complex systems. So once you're in the department, you really have an opportunity to either basically say, I'm going to be focused, I'm going to do more of the biotech part because I want to go to medical school, or you want to do more of the energy part because you want to stay in the industry and be part of clean energy, for example. The department also has a highly collaborative environment. There's some schools where it's all about cutthroat competition, who's going to be, top and stuff like that. That's not the chemical engineering department at UVA. Okay, it's a place where it's really collaborative. You get to work in teams, you get to work with each other. And I can tell you that in the real world, no one works alone. You have to be able to work through people, with people. And I think you get a strong foundation in the department. The other part that I think is very unique and I think makes UVA an excellent place is the professors of practice and the Halsey professorship. And I'm glad Michael Keane is there because he used to be a Halsey professor, now he's a professor of practice. And what you get really senior executives whom upon retirement from their company become part of the department. The Halsey professorship is a rotating thing. It's every year there's a new professor that comes in. And it's really important I think it's really impactful for the undergrads because they get to teach you what industry is about, what the real world is about, and helps you to prepare for getting into industries. And the professors of practice are similar, they're more constant, and they really help you in a way that once you get started in industry, you have a heads up on people. I think that's a really great jewel of the department. I think it makes it very unique. So I apologize for taking too long and for all the mess up in the listing, but I'd be happy to take any questions that anyone has.
Thank you, Paul. Let's see. So yeah, if there are any questions, please feel free to speak up or write it in chat.
I think something that came up in the very end there, you were talking about being able to kind of get it all in chemical engineering, the bio, the energy, the everything. Can you maybe say anything about the folks that you work with in your team, what kind of background they have, and maybe some of your colleagues that have gone into other areas?
Yeah, so obviously, when I work with people from diverse background, right? I mean, we have chemists, we have biologists, we have engineers. I think, for most of what I do and my group does is really the development of the process, not just to make small scale material, but with a line of sight to commercial, right? to scaling it up. And so because of that, that scale up requires significant engineering principles. And so we are biased towards engineering as we get into the process development stage. But we work across the board, and so we have to work with the researchers who are doing the basic biology or the basic chemistry to understand it enough, right, to be able to scale up. So I think that's the beauty of chemical engineering. It doesn't really matter what the end product is or what it does, and you can say that you worked on all of them. Whereas someone in the oncology field says, I just worked on the oncology project and stuff like that. So I think that's the difference.
We do have a question in chat. Do most people who work in the biopharma field tend to go to grad school, or can you also work with an undergrad degree?
All of the above. Obviously, depending on your degree, it's where you start, right? But once you are in, it's really your capabilities to move up. And so, someone with a bachelor's will start at a particular level. Someone with a master's will start at a higher level than that. Someone with a Ph.D. will start at a higher level. But once you are in, it's really the impact that you have. And I think it's consistent with almost every industry.
Yeah, and just to add on to that question, our bachelor's degrees from chemical engineering, I think about 40 to 50% of our undergraduates go directly into the pharma industry with an undergraduate degree. About 15 to 20% of our undergraduates go directly to graduate school. But some of those folks that go into pharma work for several years and then decide that they go back for a graduate degree.
Yeah, and the other point that I didn't make is that if you choose not to go to graduate school or wherever, chemical engineering is one of the majors where you can make quite a bit of money and be comfortable as a bachelor's student. So that's another plug for chemical engineering.
Always a good point for chemical engineering. Mike has asked a really good question, actually. Can you comment on the need and challenges to work in a global collaborative environment?
That's a tough one, especially with the differences in time zones, right, and differences in languages and stuff like that. And we've unfortunately, I think there've been some good that has come out of what happened last year, right? You can work and be effective without traveling. But still, the interpersonal connection, it's not the same talking on Zoom or WebEx than it is meeting with people. But the reality is things are global now, and so you need to be able to be prepared to work in a global environment, right? Whatever you develop is needed in China or in this, then there's competition all across the board. So I think having that global perspective for whatever the industry you go in, I think it's a critical part of it. And yes, it's a challenge, but it's also a really good way to kind of see different perspective and work with different cultures. So it's also very exciting.
I think I don't see a question right now, but I know one thing that sometimes is lacking. Could you potentially give us an idea of what a quote typical day looks like for you? Right, what you spend your time doing?
Yes. This past year has been very different than in previous years. So I'm not quite sure I know what typical means anymore. Obviously, my typical day is not like someone who has come straight out of school and doing it. I spend a lot of time in meetings, and it's meeting either to review data and provide directions, or kind of like strategic perspective of things. But yeah, one of the things that you have to do in my role is you can't just focus on the science and the development. When you meet with people, it's not all about the project and how it's doing. I mean, part of our job is also to develop people, right? And so you have to have a balance. I mean, people are here for a career, and you can't be focusing on what the company really needs. And so you have to have the balance of making sure that we are taking projects through, but also ensuring that the people who are in the group are getting what they need to be able to develop and grow and be able to get to the next level. So it's management, of people. I think it's a critical part of that, and then ensuring that the project delivers. And one of the things that I always say that the people part is probably the most important part, because the reality is we hire a lot of really, really smart people, right? And so if you focus on making sure that they are at a place where they feel like they are learning, they are growing, they're being challenged, I think the work typically takes care of itself. So I tend to bias myself as much as I can in ensuring that the people have what they need to be as successful as they can.
Thank you. So it's almost 2. Last call for questions from our audience. If not, I have one question, Paul. What's the earliest you've had to wake up for a meeting for during this COVID time?
Oh gosh, the challenge of being Central Time is even on the East Coast. No one really thinks about it, right? And the earliest has been 5 o'clock, 5 a.m. And during COVID time, I think one of the difficult things is, and working mostly from home. I've been going to the site now, working mostly from home. Sometimes I get up, I have some coffee, and I sit down here, and I realize at 2 o'clock that I haven't gotten up to eat or even brush my teeth or taking a shower, and so I got to make the time to do that because it was just so busy and exciting, and really I think everyone was working so hard, and the teamwork across countries, across sites, just to kind of make sure that we get this thing there was hugely impressive. And I used to just recently I was thinking I can reflect on 2020 and say, gosh, it was a horrible year, right? And by all measure, it was a horrible year. It was a very difficult year. But I can also look back and say, gosh, it was a year of innovation and just the ability of getting people together and seeing what they can accomplish. That value of that, I think it's really hugely important, because we would never have been able to do what we did if it wasn't for 2020. And so I balanced my perspective and not view it just as bad, but really that it was a highly innovative year as well. And so I try to balance that going forward.
Thank you, Paul. So with that, let's thank the speaker one more time and do a Zoom clap. There we go. All right. So yeah, with that, I'll close the meeting, and thank you all for being here as well.